Now we have arrived at the last disease for this lecture, trypanosomiasis. There are two types. African trypanosomiasis, or sleeping sickness, is caused by Trypanosoma brucei, and American trypanosomiasis, or Chagas disease, is caused by Trypanosoma cruzi. Both trypanosomes are blood flagellates. They are transmitted by insect vectors. African trypanosomiasis is transmitted by the tsetse fly, and American trypanosomiasis is by a triatomine kissing bug, which is found in the southern United States, Mexico, Central and South America. They are about 2 centimeters long and slightly larger than German cockroaches. Major reservoirs of infection are cattle, wild game animals for African trypanosomiasis, and humans and other mammals such as rodents for American trypanosomiasis. Since these are two quite different diseases, let's look at African trypanosomiasis first. During the first stage, the parasite is in the peripheral circulation, and symptoms include fever, headaches, muscle and joint aches, and swollen lymph nodes. In the second stage, the parasite crosses the blood-brain barrier and infects the central nervous system. This stage is life-threatening if the disease is untreated and progresses. There are two subspecies of African trypanosomes. The first causes East African sleeping sickness, which is the most severe disease and progresses faster, resulting in death within months. Trypanosoma brucei gambiense causes West African sleeping sickness, which results in daytime sleepiness and nighttime sleep disturbances. Progressive confusion and personality changes can occur. Other neurologic signs including partial paralysis, balance and walking difficulties, and eventually death within a few years can also occur. The life cycle of African trypanosomes is quite complex. Let's focus first on the right side of the diagram with the blue arrows. An infected fly injects the parasite, called trypanosomes, into the skin, which enter the lymphatic system and pass into the bloodstream. The parasites are then carried to other sites and continue to replicate asexually by binary fission. Now let's focus on the left side with the red arrows. When tsetse flies bite infected humans, trypanosomes are ingested, and they multiply in the fly midgut, undergoing some biological changes and multiplying by binary fission. They then migrate to the fly's salivary glands as epimastogotes and continue replicating. They transform into the infective trypanosomes, which are then injected into a susceptible human when an infected fly bites them. African trypanosomiasis is diagnosed by the detection and identification of trypanosomes in blood and other specimens such as lymph and cerebrospinal fluid. They range from 14 to 33 microns in size and require magnification with a microscope to be viewed. They are small kinetoplasts, dense granules of mitochondrial DNA which change position depending on the life cycle stage. They also have nuclei which are centrally located, undulating membranes, and flagellae that run along the undulating membranes and leave the body at the anterior end. You can see two blood-stage trypanosomes in the picture against a background of red blood cells. Treatment is available, but the type of medication depends on the disease stage and the trypanosome species. Some of these drugs have serious side effects as they're quite toxic, and adverse reactions can sometimes be life-threatening. Therefore, it is better to prevent infection by minimizing contact with the tsetse fly vector. This may include wearing long-sleeve shirts and pants with medium-weight material in neutral colors that blend with the background, as tsetse flies are attracted to bright and dark colors. Now let's look at the other type of trypanosomiasis. American trypanosomiasis, or Chagas disease, starts off as an acute illness with fever and swelling around the site of inoculation. Some cases may only have mild symptoms or be asymptomatic, whereas a rare number may experience severe inflammation of the heart muscle or brain. This acute phase is then followed by a chronic phase, which is generally asymptomatic, and there are few or no parasites present in blood. In about a quarter of the cases, debilitating disease can occur with heart rhythm abnormalities, dilated hearts, and sudden death or dilated intestinal tracts, resulting in digestive problems. In endemic areas, American trypanosomiasis is transmitted by the kissing bug vector. However, in Canada, transmission is mainly through blood transfusion and other blood products, organ transplantation, and vertical or congenital transmission. There were 2 cases both in Manitoba, which isn't exactly a tropical location, and both were associated with blood transfusions. There were also a case of vertical transmission from a mother who received a blood transfusion in Manitoba. It is very interesting, and since it's not the problem-based case this week, I've added the link to the paper. The life cycle of Trypanosoma cruzi is also rather complex. Let's look at the right side of the diagram with the blue arrows. An infected bug takes a blood meal, releasing trypomastigotes in its feces. Trypomastigotes enter the host either through bite wounds or intact mucosal membranes, usually the eyes, when they erupt accidentally. Trypomastigotes invade tissue and differentiate into intracellular amastigotes that multiply asexually. We're still focusing on the right side of the diagram with the blue arrows. Amastigotes differentiate into trypomastigotes that enter the circulation as bloodstream trypomastigotes. Trypomastigotes infect cells from a variety of tissues and transform into intracellular amastigotes in new infection sites. A kissing bug bites an infected human and ingests the trypomastigotes. The parasite then undergoes biological changes, and epimastigotes develop in the midgut, differentiating into infective trypomastigotes. When the infected bug takes its next blood meal, it releases trypomastigotes in its feces near the site of the bite. American trypanosomiasis is diagnosed by the detection and identification of trypanosomes in blood or cerebrospinal fluid during the acute phase of the infection. Trypomastigotes, the blood stage parasite, have large kinetoplasts. Amastigotes can be detected in tissue biopsy specimens after staining with histological stains. Because these parasites are usually absent during the chronic phase, serology is used instead. Treatment is available for Chagas disease. They're different to the ones used to treat sleeping sickness, but also have some adverse side effects, although these are not life-threatening. However, these drugs are usually not recommended for the elderly, as the side effects can be worse. To prevent transmission in endemic areas, elimination of the vector with insecticide use and improved housing is recommended. In non-endemic areas, screening of blood or organ donors have been implemented to prevent transfusion and transplantation-acquired disease. Early detection and treatment of new cases is essential in preventing congenital cases and vertical transmission.